That is what we will look at next, using Burrows-wheeler for pattern matching. Okay, just to recap, we already have a solution using suffix trees that was linear in the size of the genome and the patterns, with memory taking size of the genome for storing the suffix tree of the genome. But suffix tree, the hidden constant in this is basically C times genome, right? At most C times genome for very large genome sizes, and the C is going to be 20, right? So 20 times the genome space is required. Can we use BWT of genome as our data structure instead and then do this pattern matching, and what will be the subsequent running time? Okay, let's say that we want to search for Anna in Panama bananas, and we have already said that it occurs in 3 locations, right? This is the second location, this is the 7th location, 0 sorry, let me just write it in the computer scientist terms. This is the first location, this is 0th location, right? So first location it matches, 2, 3, 4, 5, 6, 7th location it matches, 8, 9, 9th location it matches. So if Anna is given as a pattern, then we need to output that it is present in location 1, location 7 and location 9. Okay, all 3 we have to print. Okay. If we are given the full matrix, we can just scan through it and then see that the first columns are A, N, A, N, A, N, A. And if we somehow remember the locations, we can print all of these. Okay. So, but we are only storing the first column and the last column. Okay. Can we use the same idea of inverting the BWT using the first and the last column to also search for patterns? Okay. And we'll see that the idea is actually quite similar. If you want to search for Anna, the way that we reconstructed a genome in the reverse direction, we will also search for a pattern in the reverse direction. Okay, we'll start with the last letter in the pattern A, and then we'll look at where it hits in the first column. So these are the 6 A's. Okay, and then we are going to say what is the previous letter. So that letter is in the last column, right? So then we look here and then see which ones have a N in them, right? So this has an N in them, this has an N in them, this has an N in them. So all of these are potential hits for Anna, right? Because all of these are saying that NA is present, right? So A2, N1 will be one NA that is present, A5, N2 and A6, N3 are all locations, are all 3 places where NA is occurring. Okay. So that is what is given here. The greens are the hits, the reds are the ones that are not hits, right? So then we have to only process the greens, which is A2, A5, A6 and N1, N2, N3. Okay, so now A has already been matched. Now we have to look at N1, N2 and N3, and we know that all of this will be in contiguous locations in the first column, which is this location. And in order to see if they have a in the front, then again we have to use this cyclic property and say that which of these have an A, and all of them have A's. So then there are 3 hits of A and A in this particular genome, Panama bananas, right? So that is what is going to be shown here. So then you go into, so A has been matched, N has been matched now. Now I want to match another A, the first A in the string. And basically that has also been matched now, right? So NA has been matched in 3 places. I want to now, so this NA corresponds to this NA. Okay, this has been matched. Now I want to check if this A is being matched, and that is going to be in the last column here, right? And that indeed also matches. So all of these A's are also matched, and then we basically have this A3, A4 and A5 occurring together in this particular location. Okay, so if you go back to the first column again, we have this. So we are crucially using the first to last property, right? We just have to now find the matched patterns. We need to also output the locations. So an easy way is similar to suffix trees. Initially we had a suffix tree without any locations. Now we can attach the locations. In the suffix tree we attach it to the leaves, but here we can attach it to every row in this matrix. Okay, so in addition to the first column and the last column, which is the first column is a sorted one merge, and the last column is the Burrows-wheeler transform. We will also say for each of these rows what is the position of the cyclic rotation where it starts from that row. Okay, so that will be made clearer in the next set of slides. So where are these matches located? A multiple pattern matching problem is like before, a collection of strings and a string called genome, and all positions in genome where one of the pattern appears as a substring. So where are these positions? So BWT has not revealed it so far. Okay, we have only found that in this particular case that ANA is present 3 times. Okay, but we don't have the locations yet. It will be quite convenient if somehow the locations are stored here, that this is location excuse me, this is location 1, this is location 7, and this is location 9. If for each of these rows the positions or locations are stored, then that would be quite convenient where ANA is appearing. Okay, so this ANA is position 1, this ANA is position 7, and this ANA is position 9, right? And that is indeed what we are going to do, and that is called a suffix array. Okay, so once you add the position to this first and last column of this Burrows-wheeler matrix, then those 3 columns will give you the essentially the suffix array data structure. Okay, you can think about it. So the suffix array holds the starting positions of each suffix beginning a row. Okay, so if you have this, so this is a suffix that is at 13 position. The second one is the suffix that is starting at the 5th position, right? A bananas, right? So all of the cyclic rotations, you don't consider everything that is happening after dollar. You just look at this MA bananas and all the way till the dollar. That will give you a suffix, right? And so the cyclic rotations can also be viewed as you look at all the suffixes and then you sort them. Okay, so dollar is a suffix, an empty suffix. A bananas is a suffix. AMA bananas is another suffix that is starting at location 0, 1, 2, 3. So that is attached as this 3rd location, and so on. Okay, finally S dollar is starting at location 12, right? And so that is this location here, and that is the suffix that is given here. Okay, all the bold face part of this Burrows-wheeler matrix is all the suffixes, and you know that in the Burrows-wheeler matrix they are all sorted. So you take all the suffixes, you attach positions to them, and you sort them, right, lexicographically, and that essentially is your suffix array. Okay, so this is your suffix array. It holds the starting position of each suffix beginning a row. Okay, so this 13, 15, 3, 1, 7, 9, that ordering and list of numbers is your suffix array, and then the total numbers will be, it will cover, the suffix array will have all the values from 0 to 13, because 0 will be the first suffix, second suffix, third suffix, first suffix, second suffix, third suffix, and so on until the final suffix, which is 13. Okay, all these numbers from 0 to 13 will be in some order, some sorted order based on this lexicographic sorting of the suffixes. Okay, and like I said, ANA occurs in these A3, A4, A5, and then so 179 of Panama bananas, and that will give you the exact locations where the patterns are starting. Okay, so the suffix array will hold the locations of this Burrows-wheeler matrix. So how much memory will it take? It will take 4 times the size of the genome, but as we saw in suffix trees, it will be basically 20 times the size of the genome. So we have reduced it by a factor of 5. It may not look like much in terms of recent current hardware and memory that you have in these current machines, but then, like I said, as you make it more and more efficient, it reaches more and more clinics, and you can do more and more analysis for the same amount of budget that you have for purchasing your hardware, and for the, if you're also improving your running time, for the same amount of time you're able to analyze more and more samples. Okay, so it makes the technology much more scalable. And here is the basically this slide. In 4 times the size of the genome, it is achieving what is happening in 20 times the size of the genome in suffix trees, right? And then there is also a correspondence here, in the sense that the ordering in which this is happening, right? If you lexicographically sort your suffix tree, if in your suffix tree you lexicographically sort your edges, right? So that A always comes, there is also a dollar that is not being shown here, but let's say there is also a dollar here, right? And that will be the 13th position. So you have a dollar that always comes before A comes before B comes before M comes before N comes before P, and P comes before S. Okay. If you have multiple edges that are coming out of a suffix tree, and then you lay it out in a way that each node, for the multiple edges that are branching out of that node, you lexicographically sort the edges and put it from left to right. Then if you do a left to right depth first traversal of this tree, right? So then you will hit 13 first, then you will go into this, so that is how you will go, and then you will go into this, you will go into this, so you will hit 5 next, right? Here also you are doing a lexicographic sorting, okay? Your B occurs before M occurs before N occurs before S, okay? So if you are doing that, basically you get the 13, you get the 5, you get the 3, and you are basically doing a left to right depth first search, and that is exactly the suffix array, okay? Then you will get the suffix array, okay? So this is your suffix tree whose edges are lexicographically sorted, edges at each node, and this is a suffix array. So you can see the tight link between the suffix trees and the suffix arrays. So that concludes our presentation of advanced data structures for really efficiently mapping billions of reads to a genome using just linear time, and the linear time we are also really carefully looking at the constants and making sure that you got something to the order of 4 times the number of genomes to the as the space for this data structure, okay? There are also certain other tricks and hacks and techniques, I guess, you can do to save this even further, right? So I'm not going to go into it in much more detail, but there are publications about it. You can reduce the suffix array size even further, right? We don't want to have to store all of the suffix array. We may only want to store part of it, right? So if you have a large genome, you may not want to store all the suffixes, but maybe you want to store only 1/100th of a suffix array, and that will only give you the positions of 1/100th of your suffixes. But for all the other suffixes, you spend a little bit more time to figure out what order they should come in, okay? But we are not going to go into that. These are called checkpointing, so you can even go further than 4 times the genome size. You can go 4 over 100 times the genome size, but it will increase the running time of these patterns that are being matched a little bit more, but for a shorter storage space, okay? Like I was saying, it will increase the running time by a constant factor, but it will reduce the space also by that same factor, but it is not going to touch the order notation. I mean, it will still be order of the linear in the size of the genome will be the space, and the running time will also be linear in the size of the genome. So let's just summarize or kind of remind you of how a pattern is being matched using a suffix array and the first and last columns of the Burrows-wheeler matrix. You have a pattern called ANA that you want to match in a string called Panama bananas. You look at the first and the last column, you always start reverse when you're working with Burrows-wheeler transform and suffix arrays. You start with the last letter A, and then you look at the last column and then see that the last column has A's in these positions, right? So initially you don't know where ANA is occurring, so you'll just have 0 is the starting location, 13 is the final location, so the ANA can occur anywhere between 0 and 13, okay? Now that you have seen this particular A here, right, that A is occurring in the last column, you have A1 to A6. Because of the first to last property, you know that they are the same A's here in A1 to A6. So now you have zeroed your search down to these are the positions that I have to look at, or the hits that I have to look at. In the Burrows-wheeler matrix, I have to only look at locations 1 to 6, okay, rows 1 to 6. I don't have to look at all the rows now of the Burrows-wheeler matrix, because my last letter is A, and then that letter A hits in A1 to A6, and so these are the only locations I have to look at, right? Now the next last letter is N, so A has been matched. Now I need to look at N, and out of these 6 entries, There are only 3 entries in the last column, which is the previous letter before the first column because of the cyclic rotation, right? Only 3 of them matches, and so I only have to consider those ends. The other 3 are M, B, P and B. Those are not matching. I can ignore them. So then I can go to those ends, and so those N1A, N2A and N3A are the ones that are matching these N and A here. Now finally I have the first N in the pattern, the last letter that I have to check, and those are all A's, and so your 3 and 5 are the rows in the Burrows-wheeler matrix that I have to look at. So then I have to simply look into the suffix array and then say the suffix array, which is SA of 3 to SA of 5, will give me all my hits. Okay, so this can be coded up using a simple loop where the beginning, this blue pointer, and then the ending, this red pointer, can be updated subsequently as you iterate through this loop. Here the loop runs 1 times, 2 times, 3 times and finally 4 times. Right? So the final hits are going to be, you have this suffix array that is not being shown here. So SA of 3, SA of 4 and SA of 5 will hold the positions where ANA matches the genome, which is Panama bananas. Right? And this will be number 1, number 7 and number 9. Okay, so this will be 1, this will be 7, this will be 9, which is indeed what we saw in the previous slides, if you remember it. This is 1, this is 7, this is 9, this is SA of 3, SA of 4 and SA of 5. This is your total suffix array. Okay, I hope that is clear, and that 3, 4 and 5 is what is given here. And always in terms of exact matches, you will have them always contiguous in the first row, because all these things are matching, and because this whole matrix is lexicographically sorted, so the matching hits will always be contiguous in the first column. Okay, so you only need to remember 3 and 5, and you don't have to remember 4. All the intermediate values between 3 and 5 will also be hits. Okay, similarly here between 9 and 11, the 10 is also a hit for NA. Okay, between 1 and 6, all of them are hits for the last letter A. Okay, between 9 and 11, all of them are hits for the letter NA. Between 3 and 5, all of them are hits for ANA. Okay, that is how this algorithm works. So that is like a summary of the suffix array and Burrows-wheeler transform, which is the last column of this matrix, and how using the last column and the first column and the property between them, or the correspondence between them, and how using the suffix array you can solve the multiple pattern matching problem for exact pattern matching. Okay. The final thing we are going to look at is inexact matching. Okay, so the inexact matching is we return to our original problem. That is really what we need, right? If you have disease causing mutations, then you're not going to get an exact match to your genome. You're only going to get an inexact match to the genome. But fortunately for us, inexact matching can be reduced to exact matching of a smaller string. Okay, what do I mean by that? Let's just show that by first formulating the problem. Approximate pattern matching problem is you still have a pattern and a genome and an integer d, and you want all the positions in the genome where the pattern appears as a substring with at most d mismatches. Okay, you don't want more than d mismatches. D can be greater than 0. If d is equal to 0, this will be an exact pattern matching problem. You can extend it from single pattern to collection of patterns. So you have a collection of strings or patterns, which are the short reads, and a string called genome, which is the large string, and then an integer d, and you want to find all the positions where patterns, like where a string, all the positions in the genome where a string from patterns appears as a substring with at most d mismatches. And we need to know what those mismatches are, because those mismatches are the ones that will correspond to our disease causing mutations. Or some mutations are disease causing, some are benign. Okay, so it will just point out to our mutations. that we have in our genome relative to the reference genome. Like I said, we are going to reduce this inexact matching problem to an exact matching problem, and the way we do that is, let's say that the pattern appears in the genome with one mismatch, like this, where ACT, GGCT are the same, but then the T gets changed into a A. This ACT is the same, GGCT is the same, T gets this A in the genome gets changed to a T in the personal genome. Then if you look at it, there are some substrings of the pattern, right? If D equal to 1, there are some substrings of the pattern which are intact and they will be exactly matching. For instance, this GGCT, okay? So if you divide it into two parts, if you divide the pattern equally into two parts, the first half and the second half, if we allow only one mismatch, then in any occurrence of this pattern with one mismatch, one of these two, either the first four letters or the last four letters, have to be an exact match, right? One of these two substrings has to match. So we can extend it to multiple matches also. If a pattern occurs in the genome with D mismatches, here we were looking at one mismatch, so we split it into two parts. If you have D mismatches, we split it into D plus 1 equal pieces, right? Then we can divide the pattern into D plus 1 parts, equal pieces, and find at least one exact match. So at least one of them should match. The proof goes by contradiction, right? If you split your pattern into D plus 1 pieces, and if each one of them doesn't have an exact match, then you will have D plus 1 mismatches, right? You won't have an occurrence with at most D mismatch, so that will lead to a contradiction, right? So the same way that if you have an error in the first part and another error in the mismatch in the second part, then you will have two mismatches, but we are told that it appears in the genome with one mismatch, right? The same way here, if we are looking for D mismatches, we divide the pattern into D plus 1 equal pieces, and we know that at least one of them will match exactly, right? So we split each patterns into D plus 1 patterns and then run the exact matching algorithm again. And once we find the exact hits, then we look into the neighborhood and see and kind of grow that seed into a proper matching, okay, to cover the full pattern, to see if the full pattern occurs with at most D mismatches. So this is what it looks like, right? So we have these things, and you can see here that this is one part where there is an exact match, okay? So here D is 1, 2, 3, 4, 5, 6, or the red X's are 1, 2, 3, 4, 5, 6. So we are splitting it into 7 equal pieces. So the method for seeding is that we are looking for at most D mismatches. We divide each of our strings into D plus 1 smaller pieces called seeds. We do an exact matching using the seeds, right? Check if each pattern has a seed that matches the genome exactly. If so, then check the entire pattern against the genome, okay? And this can again be done using BWT also, okay? So method 1 is you reduce, method 1 is seeding, you reduce inexact to exact matching. If D is not too large, the D plus 1 smaller pieces are reasonably large, and still this would work, okay? If D is very large, then the D plus 1 smaller pieces are very, very small, and then you will get numerous hits to the genome, okay? So then that won't work. Your seeds needs to be of a reasonable size, which means D cannot be too large, okay? This will be a problem with the other methods also, method 2 also, that we will look at shortly. But the idea is that you look at the genome, and then you get your pattern, you split it into 3 pieces. Let's say if D equal to 2, this is piece 1, this is piece 2, this is piece 3. Maybe piece 2 is hitting here exactly. Then you extend it to the left and right, and then put the pattern here, and then check if it is able to match with at most D mismatches. You check this for every place where P2 hits. For every place where P2 hits, you have to check whether the pattern can match around it or in the neighborhood of it with at most D mismatches. In this case, at most 2. Is it at most 2? Is it at most 2? You can check, right? If you're using the method 2, which is directly using BWT, You can also search for ANA with at most one mismatch, but it will lose some of the nice properties of everything staying together, right? So we'll show it here. So we are saying that we are looking for a match of this ANA, right? But if it's looking for an exact match, then we can forget about M, P, and B, right? But if we allow one mismatch, then we have to also keep track of this M, P, and B, right? Because maybe A could be occurring in the genome, right? Like that is shown here, AMA, right? So we cannot really exclude these red ones. We also need to keep them around. So the number of mismatches is 1 for MPB and then 0 for the others. And then now we need to extend all the strings with at most one mismatch, and you have to keep going that way, right? So then we'll have to look at where they are occurring. They may not be contiguous anymore in some sense. So if they are all only n's, then they will be contiguous. Now we have B, M, N, and P. In this case, these are contiguous, but these may not be contiguous in general, right? Again, you look at the number of mismatches. You have to keep that counter and carry that forward, and you can see that the P has accumulated a mismatch of 2, so you have to ignore that. You can ignore that now, and then you have you keep on proceeding with that, right? In the end, we have 5 3-mers with at most one mismatch. One is this, the exact matches are this ANA, ANA, and ANA. Then there is this AMA, right? And there is this ABA, right? So 3 exact matches and 2 matches with with one mismatch, right? So that is what is shown here, ABA, AMA, and these are all ANAs. Right? So you can take BWT and add this mismatches array to keep track of how many mismatches are there and all the places where you are hitting, okay? You cannot have just 2 numbers like we used to have before. We need to maintain all the places where it is matching, and then the mismatches that are according for all those places. The suffix array will give you the positions of these, okay? 5 is where this is located, 3 is where this is located, 1, 7, and 9, okay? So I just wanted to point out one specific thing, which is when I said you can remember these 2 numbers, 3 and 5, 9 and 11, and so on, right? This is possible in exact matching, but in inexact matching, you have to remember everything, not just 3 and 5, but also 4 or whatever else is in between. In this particular case, 1 to 6, we need to remember all of them from 1, 2, 3, 4, 5, 6, because for all of them we need to maintain what are the mismatches, okay? So you won't be able to just use 2 numbers or 2 variables to keep track of what the exact matches are. I mean, for exact matches you can use 2 variables, but for inexact matches, 2 variables, the starting and the ending row is not sufficient. You need to maintain a mismatch array like shown here, where all the hits has to be properly encountered for, and those that go above the quota of one mismatch here can be excluded, but all the others still need to maintain all the arrays, okay? All of these positions, 1, 2, 3, 4, 5. So what we have seen so far, we have come really to the concluding part. So we have seen exact matching using suffix array and suffix tree data structures to do things in linear time, and using very, very small constant using suffix arrays. And we have also seen how to extend the exact matching to inexact matching, which can then be used to figure out mutations in personal genomes. But mutations are not the only things. We know that there are insertions, deletions, duplications, and inversions. So how can we map reads in that case? So those make the problem more harder, but also, or more challenging, but also more rewarding, because this will help us reach more cases where there are deletions that will happen in certain diseases. We have already seen the case of CFTR, right, cystic fibrosis, where there is a gene CFTR where there are deletions which cause the disease. So these are important problems, even if they are harder, and there are ways in which you can handle it. And I'm not going to go into the details, but just leave the slides at that on how the reads will map if there are inversions or translocations, Inversions is when a particular genome, you have A, B, and C. This part of the genome gets inverted and you get A minus B and C. Okay. Translocations is when one part of the genome shifts to another part of the genome. All of these happens especially in cancer. So, so this is the tumor part, where large-scale genome rearrangements like inversions, translocations, and duplications or transpositions can happen. And it would be good to map reads that can also tell us about these different events that are happening in our genomes. Okay. With that, I will end the session, and thanks for your attention. And hopefully the exercise questions will help you strengthen your understanding of Burrows-wheeler transform, which in my opinion is a very elegant and a cool transform, and it really enables this suffix array like data structures for efficient mapping of mutations or mapping of reads to figure out mutations. Okay, I'll end with that, and thanks for your attention.